Oh, hello. Okay, I think we're gonna get started. So yeah, I'm really excited to have Aaron Kuan here. Aaron is an assistant professor in the neuroscience department, also biomedical engineering here at Yale. He did his bachelor's degree in physics and his PhD in applied physics, both at Harvard. So he really feels at home in a physics building, I'm sure. Among other awards, Aaron has won the Klingenstein Award, which is probably doesn't mean much to physicists, but is like one of the top early career awards in neuroscience. Yeah, I really like Aaron's work. So in the way I think about neuroscience is there's very broadly speaking two classes of things you might try to measure. One is the activity of neurons in your brain, and the other class of things would be the structure of the wiring between these neurons. And usually these are mutually exclusive kinds of experiments. So if you want to measure the activity of neurons, you either genetically modify an animal and then watch its neurons light up, or you implant electrodes and you record the electrical activity. If you want to measure the wiring between neurons, usually what you do is you first kill the animal, and then you freeze its brain, and then you slice its brain up and you do electron microscopy if what you're interested in is synapses, so the smallest little connections. So you can see how these would be mutually exclusive. You can't record the activity and have an animal dead with its brain in a freezer at the same time. Except that's what Aaron's trying to do, which I think is super cool. So I think and it involves lots of interesting microscopy, but also really cool science on top of that. So yeah, let's thank Aaron.
Thanks, Chris. And I'm really excited to be here in the physics department and have the opportunity to share some of the work that we're doing in the lab. My work is in the field of connectomics. And as Chris mentioned, basically our dream is to have sort of like a Google Maps of the brain, right? Where we have all of the structural connections and wires between all of the different neurons in the brain. And that doesn't tell you everything, right? But it gives you a really nice physical substrate, right? You can imagine, I don't know how many of you are old enough to have driven without GPS, but it was a lot harder, right? And so it's nice to have that sort of foundation. And so when you start thinking about the scales that are involved here, the big challenge is this sort of dual pressures from, on one hand, the synaptic connections in the brain are nanoscale structures. So you really need nanometer resolution to actually see them. But then an individual neuron in your brain, right, will send local wires within sort of millimeter scale. And then beyond that, they can send long-range projections to the other side of the brain, centimeters or tens of centimeters of scale, right? And so when you start thinking about, okay, I need nanometer resolution across centimeters cubed of volume, it becomes these really huge data sizes. And so that's really the fundamental challenge here, is transcending these scales. How can we kind of have our cake and eat it too, see the small stuff, but also the big picture? And for resolving the individual synaptic connections, electron microscopy has been really the gold standard for many, many decades. And you can see in a textbook like this, these beautiful images where you see the individual circular vesicles that contain the neurotransmitter molecules. And what happens when the neuron fires is that these vesicles will get dumped into this cleft, and then there'll be receptors on the other side that pick up that signal and then continue to send the message. And so you literally can see those connections between neurons. And so this has been really nice if you want to find, okay, one or two examples of what does a synapse in this type of brain region look like. But if you want to get a volume or the whole circuit, you're really limited because this inherently requires these ultra-thin sections, typically less than 100 nanometers in thickness. And so for decades, this has been kind of like a cottage industry. You have this diamond knife and this ultra-microtome machine, and you'll cut these very, very thin sections off that will float on the surface of the water, and then pick them up by hand one by one with forceps. Okay, and so you could imagine, okay, if I want a millimeter of thickness, I just collect 10,000 sections in a row without ever missing, right? And that's exactly the challenge, right? So this is now going back to some of my postdoc work. We really tried to automate as much of this process as we can to basically kind of brute force our way up the scale. Okay, so this is kind of like a semi-robotic substitution for me picking up those sections. There's a conveyor belt that's timed to the motion of this knife, and so the sections come off, they stick to this tape and come up the conveyor belt, and then this tape has these transparent slots that are in regularly spaced intervals, and they're timed to the cutting of the knife. And we can, put nice things like barcodes to keep track of which sections are what. And then in order to image this, now this kind of like, instead of an individual picture, it's like a movie reel, right? We had to convert the electron microscope into like a movie player. And so there's this in-vacuum system which contains this reel-to-reel motion, but also a fast XY stage so that when you are looking at one section, you can image many fields of view to get sort of a large area together. And this microscope basically will run autonomously for months at a time. So it basically can image the whole section, move the tape, read the next barcode, find where to find the tissue, image again, et cetera. And that's super key, that kind of level of automation when you're doing thousands of sections, right, is really the only way to get kind of that scale, kind of brute forcing up from high resolution. And so the kind of payout for this kind of technology development is what you're seeing here. So here, every frame of the movie is actually a separate image that was imaged in the microscope and then aligned back together to form this sort of sense that we can move in and out of the page. And then we used machine vision algorithms to help reconstruct the 3D morphology of individual neurons. So now this is showing single neurons. This is in the Drosophila ventral nerve cord. And you can appreciate when you zoom out, right, they have these very specific, exquisite long-range morphologies, which are very different from other neurons. But when you zoom in, you also see all these tiny little twigs that are actually where those synaptic connections are made. So this hopefully can sort of help you visualize about how you really need both that high resolution, but then you gain a totally different view when you have the large field of view. Okay, so where do we stand today? You may have, if you follow our neuroscience at all, it's been a really exciting time in our field. There have been these really milestone achievements of the fruit fly larva and adult complete connectomes. And these have been basically made possible by advances in electron microscopy, similar to what I showed here, as well as advances in machine vision and computing. All of this have sort of really made this possible. I plot this so this x-axis is time, and the y-axis is approximately number of connections and total brain volume in millimeters cubed, which scale approximately together. And it's important to point out this is a log scale on the y-axis. Okay, so from the first complete connectome, the roundworm, which has about 300 neurons, right? It was, 30 years, right? But it's actually many hundredfold in terms of size and thousandfold to the fruit fly adult. So we've come really a huge way in that time. But as we look forward and we think about bigger and bigger brains, especially mammalian brains, which are more similar to humans and ultimately human brains, it's kind of daunting because we still have these order of magnitude jumps, right? The mouse, even a mouse brain is about 5,000 times bigger than a fruit fly. And so in terms of just kind of brute force scaling, it's not just turn the crank and get the next step, right? We still have actually a lot of innovation that's going to be required to ultimately be able to do this. I think if you calculate now how many microscope years would it take to do a human brain like the fly connectome, it's something like 100,000 years or something ridiculous. So not really feasible right now. Mouse is kind of coming into view about how we might do it. And so there is some focused attention in NIH to really try to get to a whole mouse connectome and think about how we can then reach for primate and human brains. Okay, so I just want to take a moment though to talk a little bit about the impact of the fly connectome and why it's been so transformative for Drosophila neuroscience. And so in addition to having this, just the raw physical wiring between all these neurons, what you'll see in papers that analyze the connectome is they'll take these sort of neurons that have specific names, okay? And these names have been developed over many years of genetic experiments where there are specific driver lines that can label a single neuron in a fly. And you can do an experiment, you can silence that neuron and see what happens to the fly. And then you can actually match that exact neuron onto the connectome by its morphology. Okay, so then this neuron who has a name and sort of a described function, you can then jump into the connectome and see who is it connected to. And so you can really make this correspondence between structure and function because of all these advanced tools and specific information that we have about individual neurons. Okay. Now, if we try to do this in the mammalian brain, it becomes actually a lot more complicated. So this is sort of a little bit of an analogous experiment. There's basically an injection of a viral tracer from one visual cortical region and looking at the projections of those neurons into another. And this is one cell type. So, in some sense, it's as much genetic specificity as we had in the previous fly example. But you see, you get these wires from thousands of different neurons of the same type. And it's just this big jumbled mess, right? And just from looking at this, like, we can't understand what these neurons are doing, what they care about, and what their function is in the circuit. Okay, so there's sort of like a fundamentally different organization in mammalian brains, especially cortical circuits, where there are many thousands of individual neurons that work together as populations, right? And they're kind of somewhat indistinguishable from each other if you don't know anything about their activity. But when you record from them, you actually learn that they're doing something very specific. So the structure alone doesn't predict sort of very specifically what the function is. Okay, so along this theme of basically thinking about what we can learn from Drosophila connectomics and then apply to mammalian systems, there's kind of three major challenges that I want to sort of focus on. Is where we've kind of devoted some technology development thinking in the lab. So one is missing functional activity, right? We don't know what these neurons are doing just by looking at their structure. Another is the much larger scale, right? It's just kind of brute force. It's going to take too long to image these larger brains. And the third one is molecular information. So the connectome, doesn't actually tell you everything that's relevant to function, right? When you have a synapse, right, you can't easily predict the functional strength of that connection just from looking at the structure through EM. There's a lot of details such as the density of the receptors on the receiving side that make a big difference on what can happen. And so we'd really like to be able to overlay some of that molecular information onto the physical connectome as well. Okay. And so there's three thrusts in the lab that we're exploring to try to address, at least in part, these challenges. So for functional activity, what we're doing is we're going to combine in vivo measurements in the same exact brains and circuits that we do the connectome reconstruction to try to impart that functional information. Multiscale imaging, so we are using new tomographic mapping techniques with electrons and X-rays to try to get to just sort of much larger scales top-down. And then lastly, using expansion microscopy techniques to try to combine molecular-specific labeling with high-resolution imaging. Okay. So that's kind of the outline of the talk for today. Let's see how I'm doing on time. Okay. So, so this functional connectomics approach, basically how we've thought about it is that there's a quite mature field in systems neuroscience of doing these cool behavioral experiments with animals and then imaging their brains in real time. So you can sort of look at the activity of individual neurons, what they're doing while an animal is doing a cognitive task. And essentially, we want to combine that with the type of EM-based connectomics that I just described. Okay, so and this is work that started when I was a postdoc in collaboration with Chris Harvey, who's developed these decision-making tasks with mice. So these mice are trained in this virtual reality environment. They're running on a ball which controls sort of the screen, which gives them the sense that they're running in this world, which is basically just this T maze over and over again. And they learn that the color of the wallpaper, the white or the black, instructs them to turn right or left at the end of the maze. And they have to remember, so there's this delay phase where they have to actually remember, was it white or was it black? And so that makes it kind of like a non-trivial task for mice. And the reason that we do all this stuff in VR is because the mouse actually has to be stationary so we can image its brain. And so we use a technique called two-photon microscopy. And it basically takes advantage of these reporter molecules that fluoresce when the neurons are active. So this is what these dots that you're seeing are individual cell bodies that their fluorescence is basically reporting that cell, when is it active, when is it not active. And so we can identify individual neurons from each other, but what we can't see is all of the wires and branches coming out from those neurons and how they connect to other neurons. And that's basically exactly the question that matters. It's like, you know, the logic of the connectivity, we need to get to that scale, and that's what we need electron microscopy for. Okay, and so you know, this type of recording, though, has actually been really fascinating for a long time, because what we find is that individual neurons, they develop what's called a selectivity towards the decision, choice selective activity. So this is one example neuron. The x-axis is time. It's about 10 seconds for the mouse to run through this maze. And the blue trace is its average activity on left trials, and the red is its average activity in right trials. So this is a very left selective neuron. And then this is a different neuron, which is very right selective. It's basically silent on left trials, but it's really active at the end of right trials. And so collectively, these neurons give actually a lot of information about what the mouse is going to do. You can actually predict the mouse's decision based on the activity of these neurons even before the mouse makes the turn. And this is really reminiscent of this concept of decision variable, which came from theory. Basically, there's this sort of a diffusion to bound model, which you can consider like this decision variable is like the cumulative information from all of these different neurons. And when it hits a certain confidence, then the animal can make a very confident choice. So it's really kind of tempting to say, oh, maybe the decision variable is in these neurons, right? But we can't really say that until we know what the circuit is and how that information is actually produced from circuit computation. So that's basically the goal, to understand how is decision-related activity computed from the underlying circuit connectivity. Okay. Okay, and so to do this, you know, conceptually the technique is kind of simple. You do the two-photon imaging, and then you do the EM, and then you align them together, and you find the same cells. Okay. But technically, this was sort of a lot of development because of there's a big difference in the range of the resolutions of these techniques. And the EM imaging, even though we've automated, in this case, took about 6 months. Okay, so it basically, a lot of work to get what ends up still being a very small amount of tissue. This is about 1 millimeter by 1 millimeter by 1/10 of a millimeter. But that is enough to contain kind of a local circuit where we can start to look into these questions of what's the structure-function relationship. And so this is just a visualization of that data. So now each of these neurons in different colors, it's a unique neuron that has been traced through the electron microscopy data, and then they're flashing based on the dynamics that were determined from the two-photon imaging data in the behavior experiments before the animal was sacrificed. Okay, so I'll kind of jump quickly to kind of what we found. The main circuit motif that we found that was interesting is this, what we've been calling opponent inhibition. So basically, left selective excitatory neurons will activate left selective inhibitory neurons, and these guys actually have a preference for inhibiting right selective excitatory neurons. And so intuitively, this kind of gives a sort of winner-take-all type of dynamics, right? So it suppresses the opposite choice neurons, which is kind of obvious. But there's also actually an amplification at second order. Basically, once these guys are silenced, they stop activating these guys, which releases inhibition. So there's actually an amplification which comes from this motif as well. And we think that separates the activity dynamics. It really pushes into a very confident right state versus a left state as the animal is receiving kind of more and more sensory information. But we got really interested in this idea of how these motifs can support decision-making computations. And so this is ongoing work led by Mao Yasuda, who's a physics student here, and she's co-advised with myself and Shreya Saxena. And so basically, we tried to model some of these networks with recurrent neural networks. Okay, so on sort of a conceptual level, right, we have these 4 classes of neurons, right? But in reality, there's many individual neurons in each of these categories: the left, right, and excitatory and inhibitory. And you can think of there's like a range in terms of the selectivity of these inhibitory neurons, basically whether they prefer to silence the cross type or the same type, right? This is the opponent inhibition motif I just described. This is like if they had random connectivity, and here is if they actually have a preference for the same type, what we call co-selective. So there's kind of a range here. And so what we did is we set up recurrent neural networks which have inhibitory to excitatory neurons, right? This is kind of in contrast to most RNNs where basically the individual neurons, they can go positive or negative on their sign, right? So we sort of add this biological constraint. And then we trained on the behavioral data for this task. So the inputs are basically the position in this direction, the y direction, so the mouse runs at a certain speed, and then the cue, basically the white or the black cue. And then the network learns to predict the transverse direction, which is essentially reporting the decision, right? So the position and the velocity and the heading angle. And these networks, they can recapitulate this activity pretty well, which probably isn't so surprising. But what's nice about this is then this is kind of like a test system which we can interrogate much more easily than the actual brain circuits, right? We know after training we can look at all the weights and we can look at the activity patterns of each individual neuron easily. And so what we're finding is sort of there's these interesting different computational regimes which are actually dictated by some constraints that are related to biology. So opponent inhibition is in this regime, and so these plots is basically showing each type of connection, excitatory to excitatory, excitatory to inhibitory, et cetera, and then the co-selective versus the anti-selective part. So if the left side is higher, that's like to like. If the right side is higher, that's opposite. Okay, so this is sort of like the I to E that's going opposite. That's what sort of really distinguishes the opponent inhibition. So we can get networks that show this, but there's also other networks that show different regimes, like this one, which is dominated by really strong recurrent excitation and then stabilized by inhibition, which is actually co-selective on the same side. So you have these sort of like two separate circuits. And what we're finding is that those what regime you get is heavily influenced by type of constraints. For example, the sparsity of the connections, of the excitatory connections, or the fraction of the neurons that are inhibitory. And interestingly enough, when we sort of set up the network kind of close to what we see in cortical networks, so there's about 15% inhibitory, majority excitatory, with a lot of sparsity, we get opponent inhibition as opposed to the other types. So this is ongoing work, but we're really interested in kind of what are the computational consequences of these different regimes, and why did biology pick this regime in particular for this brain region. Okay, and so then more generally, I have this hypothesis that there are different connectivity rules or motifs that emerge in different brain regions, right? If you look even just across the cortex, right, you have all these different regions which are dedicated to different types of either sensory input or motor outputs or more cognitive association tasks, right? And so perhaps if we go, not just looking at this one decision region that we studied, but different sensory regions, and we might find subtle differences that kind of can key us into how that relates to what these circuits are really designed to do. Okay, so now I want to take a step back and go to this problem of scale. Okay, so one of the challenges with the experiment that I just described is that to do this co-alignment of the functional and the structural imaging, we basically had to do the whole experiment, get these 2,500 sections, image them all, align them back together, and then look for the same cells. So there's a lot of ways in which this can go terribly wrong, and you can do 6 months of imaging, and you can say, oops, we didn't quite get the right brain region. And so even though it's technically possible to image this in a reasonable amount of time, it's very kind of scary, right? And you think about, if I want to do a whole mouse brain instead of this local circuit, right, we could be doing this for 2-3 years and then realize, oh, there was something wrong in the middle of the brain and we have to start over, right? And so section thickness, even when with an automated method of imaging, still is a critical bottleneck. And so one thing we've been exploring is a way to get away from this thin sectioning without sacrificing resolution. And so this is some first preliminary work that was done in collaboration with Mark Ellisman, who has used electron tomography for many years, actually to study the nervous system. And typically, so the idea with electron tomography is essentially you can image a thicker sample, but you image at multiple tilts. And then you combine that information from those multiple tilts to then reconstruct the internal structure of your sample, right? And this is usually done, many of you may be familiar with like single particle cryo-EM, right? It's usually done to get the like highest possible quality, really detailed structure, even atomic structure of biological molecules, right? But we actually wanted to look at it a different way, which is to say, can we cheat and go a lot faster and sacrifice quality so we're not looking at like individual atoms, but we want to be able to see synaptic connections, which on that scale are actually quite large. And so we did this exercise where basically we started cutting corners. So for example, the angular range, often it'll go sort of negative 60 to 60 degrees. We started cutting that down to 30 to 20 to 15. And along with that, taking fewer images. And essentially then we were measuring, so these are little gold nanoparticles, we're measuring how much does my resolution, especially along the thickness direction, how does that get compromised as I cut these corners? And so this plot is kind of a summary of the results. Basically, the Z resolution, so the figure of merit is like with the previous technique, we're physically cutting at 40 nanometers, right? So that's essentially our Z resolution, or I guess technically it would be 80 nanometers, right? But sort of apples to apples, that's the line, right? And we can just measure from the data how that resolution improves as we include more images. And we find that actually very modest ranges of like 20 images at plus or minus 30 degrees allows us to get this same Z resolution, but for sections that are up to a micron in thickness. Okay, so that kind of like really vastly simplifies the process of what we have to go through to get these samples. And essentially we trade off a lot of physical cutting and sample handling for just more imaging time, taking more angles, et cetera. And so we're really excited with this technique because we're finding that we can do kind of the similar type of neuron reconstruction. These resolutions, again, they're like terrible compared to cryo-EM, but they're really good enough for what we want to see, right? And so we are now building, this is in the medical school basement, a new customized sort of dedicated electron tomography system. And the idea here is basically the commercial electron microscopes are really geared towards the highest possible quality of a very small field of view. And so even though we can show in the previous data I showed, we can show like a 500 times speed up, that's still not fast enough to do what we want to do. So essentially, we need a dedicated stage that basically has fast motors both in X, Y, but also in the tilt direction, and that have a large range so we can pack a lot of samples in one load and basically image overnight or a few days without changing the sample. So this is in development with support from the Wu Tsai Institute and a company called Voxa. Okay, so we think that's going to make things quite a bit easier for the type of circuit analysis, EM circuit analysis I talked about. But if we now go really top down to think about, okay, human brain, right? When you look at an image of a human brain, actually what's really striking is a lot of the space is this cabling, white matter cabling. Okay, so there's actually no synapses in these regions. It's just all of these cables connecting brain regions, circuits that have synapses, to other ones. And there's this interesting scaling law, right? As you get bigger and bigger brains, right, the surface starts to fold because you need more surface area for more circuitry, and then you need the cabling to connect those. So actually the proportion of the brain that's this white matter, these sort of like highways of information, becomes more and more, such that it's like 70% or something of the brain, of your brain is actually just cabling. And so actually the level of detail that we were talking about previously is actually unnecessary for a lot of this cabling. And so what we've been trying to do is use X-ray imaging to kind of look at this top-down view of this cabling, so that we can actually trace these cables across much longer distance, but do it faster, and then maybe combine that with high-resolution EM to give the whole picture. Okay, so the nice thing about X-rays, right, is they penetrate matter really well, right? That's why you go to the doctor to get an X-ray on your bones. And there are a whole slew of micro-computed tomography techniques that are essentially analogous to a medical X-ray. And they're really good down to about micron resolution. So you can take, this is like a whole hemisphere of a mouse brain, and you can see the individual cell bodies. But you can't really see details beyond that. So now comparing with electron microscopy, when you zoom in, you start to just see just the blobs and none of the details. And the reason of that is basically you just don't actually have enough contrast in this absorption mechanism that's used for most micro CT. So a really cool way to get around this is to use phase contrast imaging. And for high energy X-rays, the difference between the sort of the phase, so the phase shift is denoted by this delta part of the refractive index, and the imaginary part beta is the absorption. So the phase differences actually start to exceed the absorption by like factors of thousands or more when you get up to high energy X-rays. So we actually gain a ton of contrast, and so that's the basis of this X-ray nanoholography technique. So this is developed by Peter Klutens and Alexandra Pacrianu, who are collaborators at the European synchrotron, ESRF. And so, the setup of this imaging is actually quite simple. They're basically these two X-ray mirrors that focus the beam to about 10 nanometer spot size. And then as the beam is expanding, you put your sample inside. And basically, it'll do some phase shifts, and then those can be detected. So as the beam propagates, they sort of self-interfere with the forward beam, and you get these interference patterns at the detector. And so what you measure looks like this on the right, and then essentially mathematically you can convert that to what the phase should have been at your sample. So basically this is essentially Fresnel diffraction, and then once you have the phase of the sample, you use a very simple algorithm, filtered back projection. This is basically the same as electron tomography, right? To then put together the 3D structure of your sample. And the difference between doing this with X-rays and EM is X-rays you can penetrate a lot thicker, so even at high resolutions like millimeter scale. But the resolution is not quite at the level what you can do with EM. Okay, so in the X-ray imaging field, there's actually been a huge advances in the last 5 or 10 years. So we kind of went from brain imaging, seeing images like this, to recently our collaborative work. We really start to like have the sense that it looks like electron microscopy, just a little bit lower resolution, right? And so there's actually a lot of excitement that we can actually do synaptic resolution imaging with X-rays as well. And so there's development of a new beamline at the European synchrotron to essentially do just that. But what we've been focusing on the lab right now, and this is work led by Sarosh Kayani in the lab, is to look at these long-range connectivity in the white matter, because that's actually fully resolvable by X-rays right now. And so what we have developed is a protocol where we chop the brains into these pieces. These are about millimeter scale pieces. And then we can image just through them at this X-ray microscope. And so that's the video that was playing at the beginning. And that has sufficient resolution to see individual wires. So each of these tubes is an individual long-range axon that's traveling across the brain. And biology basically helps us here, right? The biophysics demand that for the conduction velocity to be high enough, these axons have to be pretty thick in diameter, and they have to have this thick insulating myelin, which is like the dark outlines of these axons. So basically, we can do single neuron tracing in the white matter from X-ray imaging alone. And so we're borrowing techniques from electron microscopy, like the automatic segmentation algorithms, to help us basically trace across these and get these sort of highway maps of the brain. So this is a view basically as if you're like in the white matter tract, right? And the structure, I mean, we're just starting to explore it now, but it's really, you kind of have this cartoon image that there are these really neat little tracks, like if you go to a server room, right? They're like bundled together, they go from one place to another, right? But here, I mean, they're all mixed together. There is a sense that there's bundles that travel together, but they actually don't travel together for very long. Individual axons will kind of travel in a bundle and then switch over to another bundle back and forth. And so trying to make sense of kind of what that structure really is, and there's really very, very little data at this resolution and scale where you're looking at actually all of the wires together and how they relate to each other and their trajectories. And so we can do things like this, which now the coloring is the principal direction of these individual axons. So you can immediately pull out, right, there's like bands that are going in this direction, there's bands that are going into the screen, kind of diagonal. And this is kind of reminiscent of if you've seen like human imaging with MRI or diffusion tensor imaging, right? They often have these kind of plots. But those are at like the whole brain level with the pixels being like a millimeter, right? With this imaging, we actually have the resolution of individual axons, right? And so when you have a bunch of axons crossing in different directions, we actually can see that, and we don't have to average them together. So this is sort of, I think, really exciting. Like to even have a map at this level of a primate brain or a human brain, it will be extremely useful for especially for understanding all of the sort of MRI imaging that we do in clinical settings. Okay, so the vision here is that, so even just doing projectome, what I just described, the sort of highways of the brain called a projectome, is really important, especially for like primates and human brains. But also it gives you the path to connect to a connectome. So the nice thing is that the X-ray and the EM, the sample prep is essentially the same. So we can actually take those chunks and then cut them for EM and get the higher resolution data. So what we think we can do is have this sort of modular connectome where you start with a full brain projectome and then you start plugging in different circuits until you're done. And then that way, if you lose a section of the brain, you're not completely hosed, right? You just say, okay, we didn't get this circuit, but we can connect up the other pieces. Okay, now for the last bit, I'm just going to introduce a molecular connectomics. And so this is, right, the idea here is that we want to have this kind of multicolor imaging that we enjoy with fluorescence microscopy, with antibody labeling, right? So electron microscopy is typically gold standard for ultrastructure, and there are some techniques like immuno-EM that you can label certain molecules, but they're really, really hard, and it's hard to do that for like a 3D volume. On the other hand, right, fluorescence microscopy, there's a whole slew of techniques, and now with super resolution techniques, you can actually get resolution that's rivaling EM. But what you usually can't do is get that high resolution and a dense context, because usually those tricks involve you have a specific label that's somehow sparse either in time or in space, right? And then that's how you beat the diffraction limit. But what we want is really not just the structures of interest, but everything around it. And so that kind of leads us to these correlative techniques, correlative light and EM, which is the previous work that I showed with the 2-photon and EM. I mean, that's one example of CLEM. And these are, they can be very powerful, but they're operationally quite difficult. And also, the light imaging is still usually diffraction limited. And so there's some ambiguity of what you're actually, where you're localizing the multicolor labeling. Okay, so pan-expansion microscopy, it basically aims to give us sort of everything that we want here. And the trick here is that we're actually just going to physically expand the sample to make it bigger and then just use light imaging to see what we want. And so this was a technique developed by Jörg Biersdorf here at Yale with his former student Anson Saad. And there are a lot of different expansion microscopy techniques. Probably you have seen some of them. I mean, the basic idea is that you use these hydrogel chemistries. It's basically what you find in diapers, right? You embed your tissue in it, you anchor what you want to image into that, and then you add water and they swell. And so there's a lot of different techniques that have been invented recently that can increase the resolution. But what's special about this approach is essentially twofold. So one is that there's a two-step expansion to get to 20x. And that allows diffraction-limited imaging to get to the kind of EM level synapse resolution scale that we actually need. And the other one is that this is it's a protocol that retains the proteins in the gel. And so then you can use a pan-protein antibody stain. So this is basically a non-specific stain for all proteins. And what happens when you do that is you get images like this, which at first it's kind of hard to see what you're looking at because the contrast is inverted, but it's actually quite similar to an inverted EM. What you find is that in the cytosol of cells, there's a lot of protein, so you get a high signal. And then in the membranes between them, there's sort of a relative lack of it. And so you essentially see the membranes from like a negative stain. And what's also nice is that at synapses, you actually see these really bright opposing parts. And that's because there's this high density of synaptic proteins. So they're specialized. Those are exactly what make the synapse work. So they just light up like this in these kind of beautiful structures. And so the kind of dream that we have now is to use this to do molecular connectomics, right? So you have the black and white. This is the pan-protein channel. But you also can do multicolor specific antibody labeling to specific molecules. So in this case, we're looking at synaptic proteins. Different synaptic proteins, they will localize at different places relative to the synaptic cleft. But you can basically, in the same sample, get both that molecular information and the structural. So Alison and Jacob in the lab have been basically trying to extend this technique to connectomics. And really important questions that we were asking, right, are do we really have the quality to actually trace these individual neuronal wires and synapses, right? And then can we scale this to the large type of volumes that we've been working with with EM? And so the first question is actually kind of a difficult thing to ask because often you don't have ground truth. Like how do we know if we're missing wires, if we're not tracing accurately? And so we actually can take advantage of this main advantage of pan expansion, which is that we can have this. So this green neuron is labeled is a genetic label. So basically, all the parts of this single neuron light up in green. And so all of the spines are green. Even if the resolution is not sufficient to see the connection of the spine, we'll see a spine out here, and we'll know that it's connected to that dendrite. So that gives us the ground truth, basically, of what all of the little spines are. And so then what we did is we blinded ourselves to that green channel. We looked only at the structural channel. And basically, we traced all the little spines, and then we compared it with the answer and saw basically what is our accuracy. And we found that basically it's close to 80% of these individual spines. And just put this in context, with traditional confocal microscopy, this is probably like 10%, 20%, right? And with EM, it's also not perfect. Like I think we're also somewhere in the range around here for EM. But EM, actually, we don't have the ground truth. So it's actually difficult to do this quantification. So this actually represents, I think, a really big step for optical microscopy-based connectomics. It really, certainly in our field, it feels like it's arrived, and it's a true possible alternative to electron-based imaging. And so we're really excited about what we can do with this approach. And so then kind of a lot of the parallel steps of how do we increase imaging throughput, how do we analyze large data sets. So we've done some pilot data sets. This is a cerebellum slice at the sort of, the scale bars are sometimes confusing here because these are the real, true physical dimensions, right? But because the brain has been expanded 20x, the scale bar is effectively 20 times smaller. But this is about 100 microns of original tissue that's been blown up to this millimeter size. And we can kind of zoom in progressively, and then we can see individual synapses coming off this Purkinje cell. So if you actually trace it out, you start to be able to get these kind of morphologies that we're used to seeing with electron microscopy. And importantly, right, in these structures, right, you'll have these axons that come by, and some of them make synapses with this dendrite, and some of them don't. And that's been the real sort of killer for light-based connectivity, is that you usually can't tell because you don't have the resolution. But here we can really see, especially because those synapses, they have those bright spots, right? We know really confidently what synapses are and aren't. Okay, and so this is again in collaboration with Jörg's lab. So Hannah designed this axially swept light sheet microscope. So this basically gives us the ability to image with much better axial resolution than confocal, and also much faster. And so this is a sort of, if you want to image quickly, we really need a fast light sheet microscope. And there are also additional kind of design concerns about loading not just one sample, but many sections of a larger brain sample. And so this is this initial design is recently published by Hannah, but we are now building a sort of a second upgraded version with support from Wu Tsai Institute that's going to be kind of really dedicated towards connectomic imaging and sort of large circuit volumes. Okay, so that's all I have for today, but I hope that I kind of gave you a sense of this landscape of kind of going top down and also bottom up in terms of brain wiring, and some ways in which these different imaging technologies can really contribute and allow us to ask new questions about how the brain structure relates to function. I'd just like to thank members of my lab who contributed to this work and many collaborators and funding, and thanks for the opportunity to speak here today.
When you were looking at these inhibitory and excitatory motifs, when you went and looked at the circuits, did the connectomics, did you find those motifs?
Yeah. So I kind of skipped over very quickly, but basically that illustration is a summary of what we found in the data. The analysis is a bit complicated because even at the scales that we're imaging, we don't get the entire circuit. So often you'll have neurons that their axons are dendrites, they leave the volume. And so we have to take a little bit of a statistical view of basically, if I'm a neuron, how many chances do I have to come near other neurons? And then what's the probability that I make a connection given if they're similar or different than me in terms of functional activity? So I didn't go through kind of that analysis, but that's published. So yeah, but we were really excited that before we did that work, it wasn't clear if at this scale you can see those types of patterns. And then so basically we saw one example, so we think, okay, this is actually a really fruitful scale to look at cortical circuits. Yeah.
In like looking at making model computational models, as you're connecting this high resolution structural with the activity, what aspects of biological plausibility do you think are most essential to include in those types of models?
That's a great question. Yeah, and that's, we're really, I think, just starting to explore that with some of the modeling that I showed, right? So I guess maybe there's two parts to that question, right? One is, what are the constraints that get us even close to a regime which is remotely biologically plausible, right? And then maybe the harder question is what are the critical parameters that really affect computation? Okay, so the first part I think is an easier question to answer, so maybe I'll talk mostly about that. But right, AI systems and RNNs and everything, right? They're very loosely inspired by biological networks, but they're very different. Not least the just distinction between excitatory and inhibitory neurons. And in biology, there are many sort of exceptions to that rule where there are neurons that are both excitatory and inhibitory. And so it's sort of an open question of why are neurons in the brain organized like that, right? I suspect there's a computational advantage or some sort of efficiency or robustness advantage, but that's something that perhaps modeling can kind of bear out. There's other physical constraints. So you have this all-to-all connectivity, right, in a perceptron. In the brain, it's really hard to do that because your neurons, as they get separated in space, you can just imagine on the first order, you're just like a spherical neuron, right? And you explore space, and it gets harder. R squared makes it harder and harder to find your partners. So because these circuits are arranged in space, you have sparse connectivity. There's basically no way around it, right? And all of the connectivity statistics are as a function of distance. And so there's a lot of ways in which the arrangement of cells in space can basically interact with their sort of connectivity statistics to give you these interesting topologies about how the networks work. So whether that's a purely kind of constraint that biology faces, or there's some sort of nice efficiencies there, I think that's also kind of an interesting question. Okay, so then moving on to that second part of what are the critical aspects, I think the big thing is plasticity and learning, right? I mean, that's kind of being borne out in the AI field, in cortical circuits specifically, where we know that even in adults, things are changing all the time. And like the circuit that we studied, for example, I think those neurons that learn to care about left and right, before the mouse has ever done that task, they almost definitely don't care, right? And you can actually, do activity imaging across the sort of progression of learning and see that those patterns emerge, right? So in some sense, a lot of what's interesting about that cognitive task is plasticity, right? And so we need to not only know the sort of sort of baseline connections, but also the rules of how they change. And those can be really subtle, right? And I think that's the challenge experimentally, is that if you just look at sort of baseline connectivity statistics, there's all these sort of homeostatic mechanisms that keep everything kind of balanced. And so you don't see them on a bulk level until you get to the level where you can say this individual neuron, this was its functional activity pattern, and there are these slight changes in its connectivity statistics with this other type of neuron. Until you get to that level, it really kind of just looks like a wash, right? And maybe that's sort of somewhat analogous with AI networks, that there's like, it's a little bit hard to understand what's going on under the box. But I think that the hope is that if we have enough sort of data with this functional and connectivity mapping, we can start to make really well-constrained theories about what those rules are. And the sort of phase space of what's possible becomes much smaller than if you're just kind of, guessing with any sort of system.
Thank you.
Sure.
Any other questions?
Yeah.
When you're going over how a given axon is like a part of in the white matter, a given axon can be in contact with with the axons of many other neurons as it sort of like goes to what it wants to project up to. Do you have any reason to believe there are like interactions between individual axons in a given bundle? However, that would happen.
Yeah, that's a great question. So I think there are a lot of developmental examples where you can have these like guidance molecules that keep axons together, or even like ones that are coming in opposite directions. They'll come and meet each other. But what's interesting though is like in mature adults, mammalian circuits where you have this like a thick white matter, right? At that point, then the axons are basically really tightly insulated from the outside world. So then they can't really talk to their neighbors. So there's sort of this dichotomy of like what happens when, and that also highlights, I think, a challenge for this type of anatomical work, right? When what we see if we sacrifice an animal, and even if we get a complete projectome map, right? A lot of that is the result of developmental processes, which we don't see, which are dynamic early on, right? So that's really only like the starting point. Then what we need to do is make theories about, okay, what do we think is happening developmentally? We need to do across time periods, or we make genetic perturbations, and we think, okay, if we mess with this this developmental process, then we predict that this wiring will get miswired. When we can actually make those predictions and see them borne out, then I think we've really kind of made progress in that. But it's like it's really an open subject, because nobody has the ability to image with a single axon resolution across the brain. So we're really trying to get there, and I think that'll open up a lot of these sort of questions. Yeah.
Actually, maybe this is a slightly related question. I've heard some people talk about learning rules that can take place in myelinated axons. I wonder if you've thought about, or if you're thinking about looking for this.
Yeah, that's a great question. So the biochemistry and biophysics of how that happens is like definitely beyond my knowledge. So, but I have, that is sort of increasingly it's seeming like the patterns of myelination can be plastic and are related to learning, right? And so I think we can take maybe a similar approach to kind of the functional connectomics that I talked about earlier, but it's challenging because I think basically the first level there's evidence that it's changing and it's plastic, but like why and then what's the sort of the initiator for that, right? If we can get a sort of clearer picture of what perturbations we can put into the system to make a sort of large change, then maybe we can start looking at that. Yeah, but yeah, that's cool.
So you were saying that plasticity is one of the kind of important variable in terms of modeling. I'm just wondering, when the mouse is given a task, do they like form a stereotypic like circuit, or like do they form like a different one to achieve the similar task?
So part of the reason that plasticity is so hard to understand is there's many different timescales and ways in which it happens, right? So there's like molecular processes that can happen on the sort of like seconds to minutes timescales, and many brain circuits are sort of organized in that way, where you just, you know, kind of like a one shot, you just reorganize something. And those kind of processes, like, we can't really see. Maybe with expansion microscopy we could see, but with EM we can't see them, right? But then these sort of like maybe longer term sort of hours to days, we do know that that information is transferred into cortex in the form of physical connections, synaptic connections. So, and those are dynamic, right? So it's sort of like there's a 1 to 3 day time scale, which they're always going up and down, but it's biased by your learning, right? And so statistically, we probably can see the effects of that type of learning if we know which neurons to look at and sort of compare. But it's sort of like a moving target, right? So I think that's the challenge.
Let's think Eric again.